Hello, my name is Professor Nick Ambleton, and I'm a consultant neonatologist in Newcastle in the northeast of England. And I'm going to be speaking today on human milk and immunity in preterm infants, opportunities and challenges. And I'd like to thank the organisers very much for inviting me today. So conflicts of interest, I did receive speaker honoraria that I donated to charity. I've received institutional research funding from Prolactin Biosciences, NeoCare, and Danone Early Life Nutrition. And I've also received funding from governmental sources, but I don't have any stock shares or personal payments at all. And we have an active research group here in Newcastle looking particularly at the microbiome in relation to feeding and nutrition studies in preterm infants. We've looked to characterize breast milk by looking at the composition of IgA, human milk oligosaccharides and bacteriophages. And we're very focused on how to prevent NEC and are exploring the role of donor human milk and fortifiers. And so when we think about milk for preterm infants, what are the major issues? Well, of course, the major concern for all of us is ensuring the babies survive. And in doing so, we want to reduce the risks of disease, particularly necrotizing enterocolitis. We want to ensure adequate growth that promotes good long-term brain outcome. And we want to do all of this in the context of recognizing that the baby is part of a family and that parents must be involved in all of the studies we do, as well as all of our clinical decision-making. And over the last 20, 30 years, we've seen improved survival of all the preterm babies. And part of this, I think, is due to better nutrition. But as more babies survive, the challenges of providing nutrition, particularly to those babies who may have had surgery, becomes more important and complex. And the scale of the challenge on a global level is enormous. Over 15 million babies are born every year who are low birth weight or preterm. In the UK, this equates to more than 50,000 babies a year who are born prematurely. And globally, deaths from complications of prematurity are the most important cause of child death. Necrotizing enterocolitis, that may seem like a very kind of niche disease just in very preterm babies, is responsible actually for more deaths in childhood than all of childhood leukemia. And the costs to the NHS of prematurity are greater than 3 billion. Of course, looking after an individual baby is very expensive. So the typical in-hospital care costs for a baby born at, say, 26-week gestation are in excess of £100,000. But survival and outcome is improving. This data on the right-hand side is being put together from national data, but just focus in that babies born at 22 weeks gestation, around about 30% of these babies now survive. And at 24 weeks gestation, more than half the babies survive. And when we say 24 weeks, what we really mean is 24 weeks post-menstrual age. In fact, these babies have only been gestating for 22 weeks. And so at less than 60% of normal in utero full gestation, most of these babies survive, and most of these will end up going to normal school. So the challenge that we face is how to optimize survival and long-term outcome. And I say optimize because increasing the amount of nutrition to babies and making them grow more rapidly may not always be in the baby's best interests. So we want to optimize growth in these babies, but also avoid disease, and particularly the disease we're scared about in preterm babies and milk feeding is necrotizing enterocolitis. And this is really the dilemma we face, how on the one hand to feed babies in order that they grow properly, whilst at the same time avoiding NEC. Now, necrotizing enterocolitis affects around about 5 to 10% of all babies born less than 28 weeks gestation. And in these babies, we recognize that both innate and adaptive immune systems are relatively immature. Most of these babies are relatively dysbiotic in terms of gut microbiome, and many of them have a leaky gut barrier, perhaps due to impaired junctions, tight junctions. They also have a smaller, thinner mucus layer. They're exposed to lower levels of secretory IgA. And they're also in a pro-inflammatory state. So they seem to have heightened TLR4 expression on the surface of the gut epithelial cells. And they also then appear to have an excess inflammatory response when this TLR4 receptor and other receptors are triggered. And there's been a lot of work recently looking at the role of the adaptive immune system and lymphocytes. And this is a very complex area, which I'm not going to go into great detail today because there will be many others in the audience who know far more about this than me. So what are the clinical risk factors for NEC? Well, there are maternal risk factors, mothers themselves who've been unwell, particularly with preeclampsia. The babies are more likely to develop NEC. Moms who've been exposed to antibiotics, increased chances. And evidence of an inflammatory response, for example, due to a raised neutrophil to lymphocyte ratio in the mother, also seems to increase the risk of NEC. But the biggest risk factor here is prematurity. So that tends to dominate all of the analysis. So degree of prematurity is the biggest risk factor for NEC. But also babies who are born growth restricted have higher risks. And we also know that antenatal steroids, where mothers have not received antenatal steroids, they're more likely to develop NEC. And in the babies themselves, babies who are unwell or with cardiovascular instability or exposed to antibiotics also have high risks, although there's just a little bit of contentious data suggesting that very brief exposure to antibiotics for the baby may, in fact, decrease your risk. And then other factors that we know reduce the risk, well, human milk is by far the most important. And the benefits are for mother's own milk, but also for donated human milk when compared to preterm formula. Babies who are systemically stable have low risks of NEC, but also over recent years, a number of RCTs, more than 50 RCTs, have shown when combined in a meta-analysis that exposure to probiotics dramatically reduces the risk of NEC. So when we talk about preterm babies, who are we talking about? Well, at the one end, we have relatively large babies. So for example, 36-weekers would be considered premature, born at 3 kilos. But the majority of our work involves these much smaller babies, as small as 22 weeks and weighing only 400 grams, with a lot of babies between these two extremes. So many babies born at, say, 28 to 30 weeks, weighing around about 1 kilo. And we're dealing, therefore, with a very heterogeneous population, some very immature babies born to mothers who themselves may have been unwell, through to relatively mature babies. And these differences are likely to impact on factors such as gastrointestinal function, but also nutrient absorption, assimilation, and the baby's metabolic capacity. So what does a typical preterm infant nutrition strategy look like? Well, at birth, we promote the use of buccal colostrum. This can be collected by the mothers themselves using a little 1 ml syringe, collecting drops of colostrum that can be then placed in the infant's mouth and allowed to interact with pharyngeal immune tissues. And the mothers can therefore themselves be directly involved in providing breast milk right from the very first few hours. We strongly promote the use of mother's own milk throughout the whole of the neonatal unit stay and also in the post-discharge period. And this is initially provided via a gastric tube. We also use donor human milk if there's a shortfall in mother's own milk. Typically, this would start at 2 or 3 days of age, once mum's lactation has a chance to establish. We use parenteral nutrition routinely in babies born less than 1,500 grams, typically for around about the first 2 weeks. And then we add a breast milk fortifier in order to promote growth in these babies, again, starting somewhere once the babies are established on milk feeds. But some of these babies will still require cow milk formula when we stop providing donor milk or if there's a shortfall in mother's milk. And babies can then start already feeding around about 32 to 34 weeks gestation. So what are the challenges? Well, there are lots of uncertainties still regarding the optimal time of when to start milk feeds and how quickly to increase. Although we're fairly convinced that pasteurized human milk is preferable to formula in reducing the risk of NEC, there remain lots of uncertainties. And particularly, we want to promote healthy gut colonization and reduce the risks of inflammation. Again, recognizing massive heterogeneity. So what works for a baby optimally at 24 weeks might be quite different at 34 weeks of age. And as I say, many uncertainties around the optimal growth rate in these babies. So faster is not always better. And as I mentioned before, we want to optimize metabolic and cognitive outcomes over the life course. So nutrient requirements in our babies are very high, and therefore the risk of poor brain development in them is also very high. Here on the left, you can see Egan Bernal, a Colombian cyclist who won the Tour de France. He consumes around about 7,000 kilocalories a day, equivalent to around about 100 kilocalories a kilo a day. Compare this to a preterm baby in whom we know the energy requirements are somewhere around about 120 to 130 kilocalories a kilo a day, and you can see that our babies require about 20 to 30% greater energy intake in this really intense energy that you expend cycling up and down mountains. And a significant proportion of that energy expenditure in our preterm babies is in the brain. Let's just think a little bit about the composition of the in utero fetus. So along the x-axis here, you have gestation. And what you can see is that minerals only represent 1 or 2% of the total weight of the baby. Lipid represents about 15% of the weight at term. But at 24 weeks, there are very few fat stores, although there are obviously lipids in brain and eye tissues. Protein represents about 10 or 12% of weight throughout all the weeks of the third trimester, which means that the major contributor to weight is actually water. So a 24-week baby is composed of approximately 90% water. Therefore, there's only about 50 grams of dry tissue. So this baby here that we had on the unit a couple of years back weighed 350 grams, and somehow he has to survive ex utero and for all of his homeostatic, physiological, and immune functions with only about 40 grams of lean tissue. And that's fairly remarkable that babies can then survive. And if we think of the energy available then within this baby who weighs 50 grams dry tissue, about the same as a chocolate bar. So the total energy in this baby, every last calorie of protein equates to about 250 calories, but only a very small proportion of that would be available for use as an energy source before you break your muscles and cardiac and liver tissues down. So preterm babies are only about 1 bite of a chocolate bar from death due to malnutrition. So nutrition is vital for these babies, obviously all of the macro micronutrients, but all of the functional bionutrients that many of you have been discussing in the conference over the last couple of days, including all of the oligosaccharides and growth factors. Very aware of the critical role of microbes, perhaps those acquired from breast milk or the environment or promoted by probiotics. And then the last aspect of nutrition that I like to consider are the technical and the socio-behavioral aspects of nutrition, which are also vital to understand. Now, brain growth in early life is very rapid, but unlike primates in whom the rapid period of brain growth happens before term, the rapid period of brain growth in humans occurs in the third trimester and the first 2 years of life, during which time we acquire about 85 to 90% of our final adult volume. And unlike many other mammals, we actually have a very large brain. So in the rhinoceros, the brain only occupies a small proportion of the head size, whereas in humans, it's the entire cranial cavity. And when you plot this out on a logarithmic scale, you can see that most mammals plot out somewhere on that average line, but humans are off the scale. And compared to most other mammals, the brain of a human is about 10 to 20 times as big. And everything about breast milk composition is designed to optimize brain growth in these babies. And there are clearly lots of different processes happening during this time period when there's a rapid brain growth, neuronal migration, apoptosis, synaptogenesis, and myelination. And increasing evidence that many of these processes are under the control of IGF-1, and again, emerging data showing that different macronutrient intakes impact on the levels of IGF-1. And just to show you how dramatic nutrient intakes might be, this data shows you the amount of protein and energy that babies receive just in the first week of life as a very premature baby, with their developmental quotient 18 months later. And what you see is that for every extra 10 kilocalories a kilo a day that these babies received just in the first week, they have a 4.6 advantage in their mental developmental index at 18 months of age. And for every extra gram of protein, this was an 8-point advantage in MDI. So a massive impact on later brain development from just macronutrient intakes in the first week of life. This is very long-term data from Alan Lucas's cohorts, where they compared a standard milk diet to a high milk diet. And what you see is that 16 years later, babies on a high macronutrient intake have an 8-point higher verbal IQ compared to those on a standard diet. Now, this didn't result in any difference in brain volume, but they could detect differences in the caudate nucleus. So 16 years later, we can find MRI evidence of how we fed these babies milk just in the first 4 weeks of life. And this and many other studies has resulted in a paradigm shift where we start to recognize that milk is really very much more than food. Now, this is how I used to think of milk nutrition when I first started as a junior doctor, that you put milk into this long tube, you suck out the fluid and nutrients, and you produce waste product at the other end. But of course, a lot of work in the last 20 years has explained to us the critical role of metabolites produced, but also the immune and hormonal impacts and different microbial structures and processes going on. And breast milk probably is best thought of as a developmentally regulated maternal infant biochemical signaling pathway, emphasizing that nutrition is much more than nutrients and breast milk is more than food. Now, what's the impact of breast milk? So here they took adolescents who were 15 years old, and they showed that the largest predictors of later IQ were social class and breast milk exposure. So the amount of breast milk you had as a preterm baby was associated 15 years later with your IQ and white matter volume on MRI. And more recent MRI studies have supported this. So in this study of 68 premature babies, 40 human milk fed and 24 formula, they did MRI at term and were able to show larger brain volumes, but also larger amygdala, cerebellum, and also better white matter organization on diffusion tensor imaging. In this study of James Baldwin's group, they took 47 infants, and they looked at those who received exclusive breast milk for more than 3/4 of the day, and they showed in these babies greater connectivity, particularly in the corpus callosum, corticospinal tracts, and the internal capsule. And the impact of these better connections was even more dramatic when they looked at those babies who received breast milk for more than 90% of their days on the neonatal unit. Long-term studies confirm this. So this is data at 7 years of age of preterm babies born less than 30 weeks, and they looked at the number of days when breast milk represented more than half of the milk intake. And what they will just show was better performance, IQ, working memory, and motor function 7 years later related to the amount of breast milk they had. So that for every extra day that babies received breast milk intake, more than 50%, the IQ was 0.5 points higher. So a lot of studies to support the use of mother's own breast milk, but these advantages in the brain do not apply to donor milk. So 2 large studies recently have shown no neurodevelopmental advantage to donated human milk. And that might be because pasteurization partially inactivates some of the functional components. So human milk improves survival and outcomes through reducing NEC and sepsis. There's a direct impact of milk nutrients on the brain and an interaction with the immune system and lower rates of NEC. And there are many studies now that have looked at different components in human milk or bovine milk and their impacts on gut microbial metabolism, which may result in a whole range of different metabolites, nutrients. So for example, amino acids and short chain fatty acids, but also production of signaling molecules through the gut-brain axis. So for the last 10 years, we've set up a biobank where we were collecting samples that otherwise would have been put in the rubbish bin. And this work's been led by Janet Berrington and supported by Chris Stewart and many others, where we salvage samples from babies born less than 1,500 grams. And we've used this salvaged breast milk to study the impact of human milk oligosaccharides, but also the microbiome, the virome, to do metabolomic analysis, but also we've now moved on to salvaging blood samples from the lab and salvaging gut tissue that may have been resected for clinical reasons. And we take parents' consent to retrieve some of that resected tissue and use it for the development of organoids. And all of this is covered by the Great North Neonatal Biobank. So here's an example of some of the work that we've been doing. This was led by Andrea Massey. So we used our biobank to identify 33 babies with NEC, 37 matched healthy controls. And what you see on the left here is that the maternal milk oligosaccharide profile was very different in the red for the babies who then developed NEC compared to controls. And when they looked to see which individual HMOs were explaining that difference, a lot of it seems to come down to this single HMO DSLNT, where the babies fed milk from mothers with very high levels of DSLNT had a much lower rate of NEC. And on the right, you can see this data presented in a box plot. So in green are the babies who did not develop NEC, and in red are the babies who did develop NEC. And you can see that these two groups separate fairly well. And this level of DSLNT also seems then to impact on the microbiome. So here you see babies with high levels of DSLNT. Each of these circles represents a different time point over the first 60 days. And what you can see is that babies moving from preterm gut community type 1, which is dominated by Staphylococcus, through to preterm gut community type 5, which is bifido dominant. This is the pattern in the babies who remain healthy. But this is the pattern in babies with low levels of DSLNT, and you can see that they don't seem to progress through to the bifidobacterium-dominated microbiome. We've also used our samples to look at the association with secretory IgA. So here we took 34 babies with NEC and 44 control infants. So in the box plot here, you can see the levels of secretory IgA in the breast milk in the babies who remain healthy over the first 40 days of life. And then superimposed on this are the babies who developed NEC, and you can see that in the first few days, babies who go on to develop NEC are being fed with milk that has much lower levels of IgA. And this continues over the first few weeks of life, highly significant differences in the amount of IgA between the babies who develop NEC and those who remain healthy. So we were interested to explore whether avoiding human milk might be good. We know, as I say, that the microbiome shifts, and the question we wanted to ask in this RCT was whether an exclusive human milk diet impacted on the microbiome. So we took 120 babies who were randomized to an exclusive human milk diet with mother's own milk, but supplemented with donor human milk and a use of a human milk fortifier from Prolacta, compared to control babies who had a shortfall made up with formula and they used a bovine fortifier. And we collected samples from 116 infants, and on the left here you can see the time points when we studied these babies during their neonatal stay. But contrary to what we expected, we saw absolutely no impact of an exclusive human milk diet on microbiome richness or diversity. So we concluded that if an exclusive human milk diet was associated with improved immune function, the effect does not appear to be modulated through the microbiome. And we're also able to use this study as an opportunity to look into lymphocyte maturation. And so this is currently unpublished data, but we looked at the different lymphocyte and immune clusters. And on the right-hand side here, you can see each of these lines represents a different infant sample, or on the right, an adult sample. And you can see, maybe not surprisingly, that babies, the preterm infants, have much lower levels of CD4 and much higher levels of naive CD4 cells. So very, very different lymphocyte profiles in these babies. But we weren't able to show any real differences overall in the effect of the exclusive human milk diet. And we then able to look at how the lymphocyte clusters change over time. So on the left-hand side here, you can see 14 different lymphocyte clusters. And in mauve, we have the samples taken when the babies got to full feed. So this is typically around about 2 or 3 weeks of age. And then the red box plot on the right is when we finished the study. And overall, there aren't very many differences except for a reduction in activated T regulatory cells as the study progressed. And on the right-hand side here, you can see how the pattern changes in the heat map. So on the left of the heat map, you can see the pattern differences in the clusters when the babies reach full feeds. And again, the pattern when the babies were at the end of the study. So you can see dramatic changes over a relatively short period of time in these babies. So we're still left with the question of whether human milk might reduce the risk of NEC, and if so, how does it do it? Well, this is data from a Cochrane study we published 3 or 4 years ago now, where we show that the risk of NEC is dramatically lower if the babies receive donated human milk. And this has been supported by a study that isn't yet published, but from the States, 480 babies randomized to either using donated milk or formula if there was a shortfall, and they showed no difference in brain function at 2 years of age, but a halving of the risk of NEC in the babies receiving donated human milk. And I think given that evidence in the Cochrane, it's now challenging to consider whether we can conduct further randomized controlled trials of donor human milk versus formula. So we're faced with a number of challenges. Here are some of the clinical challenges. How do we best promote growth without causing disease such as NEC, and how do we optimize the growth pattern for long-term health? And there remain uncertainties about when we should start and increase milk feeds, what milk we should give and for how long, and when or perhaps if we should actually start fortifying. And the heterogeneity of the babies that we look after should encourage all of us to perhaps think about a more personalized approach to how we practice medicine. There are a number of research challenges, so the heterogeneity also perhaps limits the generalizability of some of the randomized trials and the systematic reviews. Biologically, it's very difficult for us to study NEC because it's virtually impossible to get access to the actual gut tissue unless the babies are unwell enough to need an operation. There are obviously a series of ethical issues around blood sampling in babies who are so small, and the burden that we might place on parents by asking them to contribute to our research studies. And then when we think about the interventions we want to study, we need better characterization of some of the bionutrients that we want to use, such as the different types of bovine lactoferrin formulations that might be available. And again, some of the challenges we face in probiotic studies, such as whether effects may be at the species or the strain level, and whether the effects are different depending on the milk matrix into which they are added. But there are lots of opportunities here. NEC is a major contributor to mortality and serious morbidity in these babies, and so a better understanding is going to save lives and save money. There are lots of bionutrient supplements, many of which may not be that expensive, which are likely to impact on immune function where we might want to add them to the milk feeds. And there are also issues here for donor milk usage. Perhaps we might be able to identify donor milk which has the highest density of DSLNT and direct this to the babies at highest risk of NEC. And we also need to better understand how we can promote and support continued use of mother's own milk. So I'm going to finish there. A very quick whiz through some of the issues that we face as neonatologists. I would like to thank all of these people who've conducted research and supported us, as well as all of the funders for our studies. But most importantly, thank the parents for their willingness to support our research over the last 10, 20 years. So I look forward to taking any questions. Thank you very much.